Very good afternoon to one and all present over. On behalf of Mehsana Urban Institute of Sciences, Ganpat University, I, Miss Melisha, would like to welcome you all on our third day of 1 week advanced level breach courses organized by Department of Microbiology and Department of Medical Laboratory Technology. For today's session, we are privileged to have with us Dr. Snehal Rami, Coordinator, Department of Microbiology, MUIS, Ganpat University. Dr. Hardik Shah, Coordinator, Department of Medical Laboratory Technology, MUIS, Ganpat University. Dr. Damodar Gupta, Scientist F from DRDO. Our esteemed speaker for today's session is Dr. Sweta Sanguri, working as Associate Scientist, Clinical Research at Friends Life Science. We also have with us the coordinators of various other departments, faculty colleagues, and several registered students from various institutes across India. Now, not taking much of your time, I request Dr. Ritesh Arya to introduce our today's speaker, Dr. Sweta Sanguri. Please, sir.
Yes, thank you so much, Miss Millie, for a nice introduction. Today we have Dr. Shweta Sanguri with us. She is a skilled scientist in the field of life sciences with a specialization in molecular biology of normal and cancer cells. Her expertise lies in studying the intricate working of cellular mechanisms, particularly focusing on understanding the complexities of the biological processes. In her current profile as an associate scientist in clinical research at Stanley Cohen, Dr. Shweta upholds a strong dedication to maintaining ethical standards and regulatory compliance. She is she has comprehensive understanding of good clinical practice, guidelines, and the regulatory requirements involved. Thriving in the fast-paced research environment, she remains adaptable, quick to learn, and committed to professional growth in the field of life sciences. Dr. Shweta possesses a remarkable educational background, having obtained a postdoctorate and a doctorate in life sciences, specializing in molecular biology of normal and cancer cells. Her research expertise lies in investigating the intricate role of TLR signals exposed to heavy ion radiation alongside comprehending the mechanistic intricacies of radiation response in radiation protection and mitigation. Complementing her advanced degrees, she upholds a MPhil in life sciences, master's in biotechnology, and bachelor degree in biotechnology honors. Noteworthy accomplishments including achievements in All India rank 28 in CSIR NET JRF examination in life sciences and an All India rank 245 in GATE examination in biotechnology. Her extensive educational foundation has enabled her to acquire profound knowledge and technical proficiency in the field of study. She has made significant contributions to the academic community through various means, including delivering invited talks and presentations, participating in workshops, online schools, and publishing research articles. Her research work has been published in reputed journals such as Frontiers in Oncology, BMC Cell Biology, and Journal of Tumor Medicine and Prevention, showcasing her expertise in areas such as radiation biology, tumor microenvironment, and the role of toll-like receptors in cancer biology. This publication highlights her ability to conduct in-depth studies, present novel findings, and contribute to the existing body of scientific knowledge. In addition to her publications, Dr. Shweta actively participates in conferences and scientific meetings where she has presented her research through short communications and invited talks. Dr. Shweta's knowledge and expertise in life sciences, molecular biology, and clinical research make her an asset to the scientific community. Today, through her engaging talk, She will inspire and empower students, contributing to the growth of future scientific leaders. We also have today Dr. Damodar Gupta from INMAS, that is Institute for Nuclear Medicine and Allied Sciences, which is a institute of DRDO, Government of India, Ministry of Defense. Sir is working as a senior scientist and I think it is really very difficult for me to explain his achievements just in a few couple of minutes. He did his PhD from Chaudhary Charan Singh University, and uh, it gives me immense pleasure to inform that Dr. Shweta is a student, PhD student of Dr. Damodar. So it is really very great movement that a student is representing in front of his guide. So I think Google is the best source to know Dr. Damodar Gupta's achievements. We have Research Scholar and uh, Google Scholar and ResearchGate. You can see his achievements from contributions to the scientific communities is really outstanding. He has very good experience of postdoctorate from United States and many more achievements are there. I think it is really very difficult for me to explain his achievements. Even an hour, 2 hours might be not sufficient to explain his achievements. So not wasting much of our time, I welcome both of them on this digital platform and I greet them on behalf of Ganpat University. And I request Dr. Shweta to share his presentation and start her talk.
Thank you so much, Dr. Ritesh, for such a nice introduction. My name is Shweta Sangri, and I am an associate scientist with Strand Life Sciences, Bangalore. And I am delighted to have this incredible opportunity to speak before all of you today. And I want to express my sincere gratitude to the organizers and Dr. Ritesh for providing me with this platform to share my knowledge and passion for genomics. So in this presentation, which is titled Genomics and Advanced Sequencing Techniques, an Introduction to Precision Medicine, we'll explore the cutting-edge sequencing techniques that have revolutionized our understanding of the human genome. Additionally, we will also discuss the emerging field of precision medicine and its profound impact on healthcare. And by the end of this lecture, my aim is for each and every one of you to gain a solid grasp of the fundamental concepts of genomics and its potential in shaping the future of medicine. So once again, thank you for having me today. Let's make this genomics lecture a captivating and enlightening experience. So let's start with what a genome is and how does it look like. So the complete set of genetic material within an organism, it's called its genome. So the genome will include all the genes, the non-coding region, and the regulatory elements. So our genome is like a fancy term for all the DNA in our bodies. So it's not just humans, all living organism, whether it is a beautiful flower or it is a fascinating animal, they all have their own genome. So this genome contains all the information which is needed to build and maintain each organism throughout its life. So it's like an incredible operating manual that guides our growth. It helps our organs to function properly, and it also occasionally repairs the damage that our body sustains during our growth and our lifetime in Earth. So what makes our genome even more incredible is that it is unique to each one of us. The more we understand our genome and how it works, the better we can grasp our own health and make informed decisions to take care of ourselves. So the basic fundamental building blocks of DNA. DNA is composed of 4 bases. All of us here know about adenine, thymine, cytosine, and guanine. So these bases, they pair with each other in a specific manner. A will always pair with T and C will always pair with G. So the sequence of these bases forms genetic code that carries the instruction for building and maintaining an organism. And multiple bases together form a DNA molecule. And these molecules are organized into structure which are called as genes. Now, genes are specific segments of DNA that contains the instruction for producing proteins. These proteins which are essential for our structure, function of our cells, and each gene carries specific information which is needed to synthesize a protein or a functional RNA molecule. Now, in eukaryotic cells, including us humans, DNA, this is packaged and organized, and it is packaged and organized with proteins called histones. We must all have heard about histone proteins. So this DNA is tightly packed with 8 histone proteins. And after this tight wrapping, this packaging helps the DNA to get condensed. And this condensation and packaging also helped in gene regulation by making certain genes more accessible to transcription and certain genes less accessible to transcription. Depends on the cell type, the cell in which the tissue in which the cell belongs, or the organ type. Depending on the cell's condition, the gene expression changes. So nucleosome, this nucleosome is the condensed form of DNA with histone protein. It is further condensed and it folds to form a high order structure, which is called as chromosome. We all have heard about chromosome. We know what chromosome is. So they contain many genes and they play a crucial role in maintaining the integrity and organization of genetic material within a cell. So if we stretch out all the DNA from a single human cell, it will form almost a 6 feet long strand. And it is hard to believe that all this DNA can fit inside such teeny cell nucleus, of which we can only see with a specific or with a special microscope.
Now, what is genomics? We just discussed what a genome is, and now what is genomics? The genomics is the study of organisms' entire genetic blueprint, which is called as genome. So this genome is like a book that contains all the instructions that are needed to build and operate a living organism. So just like each chapter in a book tells us a unique story, different section of the genome, which are the genes, it hold instructions for specific traits like the color of our eye, or our susceptibility to a specific disease, say cardiovascular disease, or a predisposition to a disease like diabetes. So this all information is present in our genes. But there is more to the story than just the genes. So there are other parts of the genome called as non-coding regions, and they were previously considered junk DNA. However, we now know that these non-coding regions actually play an important role. So, however, we know that these non-coding regions actually play an important role by controlling how genes are turned on and how these are turned off. So by studying genomics, we can uncover the secrets of the non-coding regions as well and understand how they shape the diversity of living things and they impact our health. So advancement in genomics, it enables us to identify genes, discover genetic variations, and understand how they impact health, disease, and the evolutionary process. And genomics has a wide range of applications that touches our life. So I've tried to get few of them here. So like in agriculture, genomics aid in breeding crops and livestock with the improved traits such as higher yield or disease resistance. Genomics also contributes to environmental conservation by studying the genomes of endangered species and understanding their unique adaptation. It can help us discover why some people get sick from certain infections, environmental conditions, and behaviors. Like during the COVID outbreak, we have seen that some of the healthiest people, they passed because of the disease, and someone who was already with comorbidity or was not very healthy in the beginning survived. So why this kind of discrepancy? And like, for example, like there are people who exercise their whole lives, who eat a healthy diet, they have a regular medical checkups, but still they die of a heart attack at the age of 40. So, and there are also people who smoke, who never exercise, they eat unhealthy food, but they still live to be 100. So genomics may hold the key to understanding these differences, understanding more about disease, which are caused by a single gene, which is the genetics, and complex diseases, which are caused by multiple genes, along with the environmental factor. This is genomics. So both genetics and genomic study, it can lead to earlier diagnosis, it can lead to interventions and the targeted treatments. field of genomics has undergone remarkable transformation thanks to the pioneer techniques that have shaped our understanding of intricate word which is encoded within our DNA. So DNA cloning is a technique which was invented by Stanley Cohen and Herbert Boyer in the year 1970s. It revolutionized the field of genomics. So their groundbreaking work in recombinant DNA technology, it enabled the cloning and manipulation of specific DNA fragments. It provided researchers with the ability to study genes and their functions. In 1977, Frederick Sanger developed the first method for DNA sequencing to determine the precise order of nucleotides in the DNA. So sequencing is to know how the nucleotide bases, how A, T, G, C, how they are sequenced, how they are present in a segment of DNA. This is sequencing. And Frederick Sanger, he developed the first method for DNA sequencing. And this technique, Sanger's technique, is called as di-deoxy sequencing. We'll discuss it later. This led to the foundation of large-scale DNA sequencing, and it significantly contributed to the Human Genome Project. And we will talk about Human Genome Project too. Another transformative technique, polymerase chain reaction, PCR, was invented by Carrie B. Mullis in 1983. It provided a simple and efficient method for amplifying specific DNA sequences, revolutionizing the way genetic material is analyzed and studied. These techniques have played a crucial role in advancing genomic research, enabling significant discoveries and advancement in understanding the human genome and molecular biology as a whole. So in the world of genetics and molecular biology, understanding the fundamental building block of DNA is essential. A closer look at the structure of DNA shows that a DNA, like a deoxyribonucleotide triphosphate chain, it is made up of 1-deoxyribose sugar, a base, and a triphosphate. I'll show you another slide to make this a little more clear. So this is a nucleotide, a nucleotide triphosphate. It has a deoxyribose sugar and a base. Base can be anything, adenine, guanine, cytosine, or uracil. Depends on what is the complementary base. And these are the 3 triphosphates, which are also the part of nucleotide. Let's look at the pentose sugars as well, because they are also very essential part of a nucleic acid, both RNA and DNA. So ribose sugar, this is a 5-carbon sugar which is present in RNA, ribonucleic acid. It plays a central role in the structure and function of RNA. It has hydroxyl group attached to both the 3rd carbon and the 2nd carbon. Like we can see the OH group attached to carbon number 2 and carbon number 3 in ribose sugar. Now, if we talk about deoxyribose sugar, deoxy means less, 1 oxygen less. So it has 1 oxygen, 1 hydroxyl group less than the ribose sugar at the 2' end. So at 2' end, rather than having an hydroxyl group, DNA sugar, the deoxyribose sugar, which is present in DNA, has hydrogen attached to carbon number 2 in place of hydroxyl group when we compare with the ribose sugar structure. And another important variant of ribose sugar is di-deoxyribose. Here, both the hydroxyl group at the 2' and 3' end have been replaced by hydrogen only. So it is deficient in 2 oxygen atoms than the original ribose sugar, hence the name di-deoxyribose. So now we know about the structures of the ribose sugar. Now a little bit about DNA polymerase. So DNA polymerase is an enzyme which helps in DNA replication. It synthesizes new DNA strand. It based on the existing template. So it needs a template, and based on the nucleotide sequence on the parent template, it adds on 1 nucleotide at a time, and specifically in the 5' to 3' direction. So the 5' to 3' direction, it refers to the carbon atom in the deoxyribose sugar, the structure that we just saw earlier. I'll go back once. Maybe forward here. So we can see the carbon atom 2 here, and this is the carbon atom 5. phosphate group is attached to carbon 5. And carbon number 3, there is a hydroxyl group in deoxyribose sugar. So this is the structure of deoxyribose sugar. And because of this specific structure and orientation of DNA polymerase can only add new nucleotide to the 3' end of the growing DNA strand. So this is the DNA polymerase. This is the incoming nucleotide. The 5' phosphate, which is attached to the phosphate group, which is attached to the 5' end of the ribose sugar, it attaches with the 3' hydroxyl group to form a phosphodiester bond, which forms the backbone of DNA. So this bond is called phosphodiester bond. And for a phosphodiester bond to form, both 5' carbon and 3' carbon are very important. I hope it is clear from this diagram here that carbon atom 3 and carbon atom 5 both are important for this phosphodiester bond to form, for DNA polymerase to add one new nucleotide, and for the DNA chain to grow. So as DNA polymerase add each nucleotide, it catalyzes the formation of phosphodiester bond between the 3' end of growing DNA and 5' bond of 5' phosphate group of the incoming nucleotide. So this process results in the formation of a continuous DNA strand. But what happens when a regular deoxynucleotide triphosphate is not present? This is a dideoxyribose sugar or dideoxyribonucleotide. Here, the 3' oxygen group is absent. It is deoxygenated both at carbon 2 and carbon 3 position. So when a dideoxynucleotide triphosphate is added to the strand instead of a regular DNTP, the absence of the oxygen atom at the 3' carbon position, it prevents the formation of phosphodiester bond with the next incoming nucleotide. So this is the next incoming nucleotide, and DNA polymerase will not be able to act because there is no hydroxyl group at the 3' end, and the chain will terminate. DNA polymerization will terminate here with and the last nucleotide will be the dideoxynucleotide here. So this leads to the termination of DNA chain at that specific position. So the deoxyribose sugar backbone along with the phosphate group, it forms the structural framework of the DNA double helix. And the incorporation of dideoxyribose sugar in the sequencing reaction prevents further elongation of DNA strand, allowing for the determination of precise nucleotide sequence. So we are talking about dideoxynucleotide because now we are going to discuss about the Sanger sequencing method and how it utilizes dideoxynucleotide for DNA sequencing. So DNA sequencing, Sanger sequencing technology is also known as chain termination method. It revolutionized the DNA sequencing, and it played a crucial role in deciphering the genetic code. So this process involves several steps, and it allows for the determination of precise order of nucleotides in a DNA molecule. So while newer sequencing technologies have surpassed Sanger sequencing in speed and in throughput, this still remains a valuable method. So like a lot of us can say, like, because next generation sequencing is available, NGS is now available, third generation sequencing techniques are available, why are we still talking about Sanger sequencing? But this is really important. If you want to know what genomics is about the other sequencing technology, this is the gold standard. So here, before going to the next generation and the third generation sequencing techniques, we will talk about what Sanger sequencing is, because it still remains a valuable method of small scale sequencing projects, validation of sequencing results, and targeted sequencing applications. So in this process, the DNA sequence of interest, it is used as a template. It can be derived from a tissue or a cell. So DNA is isolated. And the DNA primer and the buffer, they are heated to 100 degrees. And this heat separates the DNA into single strand. The mixture is now cooled to 67 degrees, which allows the sequencing primers to bind. Now I want to add here is because this technique was invented much before the PCR technique, and Taq polymerase was not yet available. So the DNA polymerase that was used in this process was thermolabile. It was not thermostable. So after heating it, at every step we have to add DNA polymerase. In PCR, what happens with the invention of Taq polymerase, it can withstand any temperature. We can heat it to 100 or then cool down to 45, 60. It still is active. But the Sanger technique, it was invented much before the PCR method. So here, this is a very labor intensive method. The original Sanger method is very labor intensive. So after this mixture cool downs to 67 degrees centigrade, then we add our DNA polymerase and radio labeled one of the nucleotides. That time fluorescent tags were also not available, and this method utilized radioactivity for sequencing. So then a total of 4 tubes, one for each dideoxynucleotide triphosphates, are used like this. And the structure of dideoxynucleotide. So in DNA polymerase, all 4 DNTPs, ATP, GTP, CTP, and TTP, they are added in the 4 tubes, and 4 separate dideoxynucleotide triphosphate is added to these tubes. And now dideoxynucleotide triphosphate is incorporated into the strand, terminating the fragment. So the dideoxynucleotide triphosphate is added at a very lower concentration compared to the normal deoxynucleotide triphosphate, so that this incorporation is random. So in each tube, there will be different fragments of different length. All the fragments in each tube will start with the same primer, because we are using same primer in all the 4 tubes, but they will end in the same nucleotide in one tube. So in the first tube, if we added dideoxynucleotide triphosphate, so all the fragments of different length will end up with dideoxyadenine triphosphate. Similarly, in the second tube, the starting fragment will be the primer, which will be the same, and the terminating fragment will be the other dideoxynucleotide. So these 4 sequencing reactions are then mixed with a loading dye, the next step of the Sanger sequencing method.
Each reaction gets loaded in a separate lane of a polyacrylamide gel. The fragments move through the gel at different speeds depending on their size. So the smallest one will move fastest, and the DNA is then transferred to a membrane, and the radiation from DATP in the fragments get detected with a X-ray film. This results in bands showing for each fragment, and the DNA is read from 5' to 3' to call the bases. So we start with the shortest fragment first, and we follow this again and again, so we have a sequence ready which is complementary to our template DNA. Now the original Sanger sequencing, as already discussed, is a very labor intensive process, and sequencing 200 nucleotides from only a few samples used to take 4 days earlier. And Sanger sequencing, it provides 1 read per sample. It covers only 800 to 850 bases. NGS, if we compare, in contrast, can generate billions of read per sample and almost up to 16 terabases, which allows sequencing of 128 human genomes in a single run. Now, why use Sanger sequencing when there is NGS? Sanger sequencing, despite the availability of new technologies like NGS, remains the gold standard for sequencing. It is highly accurate. The base calling accuracy is 99.9%. And for smaller samples, Sanger sequencing is more cost effective and faster. However, for larger sample numbers, NGS offers cost effectiveness and a quicker processing. So for fewer than 20 samples or genes to sequence, Sanger sequencing still remains the method of choice. And for larger samples or extensive sequencing need, NGS offers practicality and efficiency.
Now that we have discussed a bit about Sanger sequencing method, we'll go to the Human Genome Project. Now, what is Human Genome Project? So it was an international scientific research project that aimed to map and sequence the entire human genome. So this was a collaborative effort which involved researchers from around the world, and it was conducted between 1990 and it was deemed complete in 2003. So the human genome, it refers to the complete set of genetic information which is present in human. It includes all the genes, which are the segments of DNA which code for protein, as well as the non-coding regions of the DNA that have regulatory functions. Now, the main goal of Human Genome Project was to determine the sequence of approximately 3 billion base pair. So that makes up the entire human genome. So to identify all the genes within the genome and to understand the structure and the functions of these genes, by studying the human genome, it was aimed to gain insight into human biology, health, and the diseases. Now, Human Genome Project, it was completed ahead of schedule, which was 13 years. Within 13, it was estimated to be completed in 15 years, but it took 13 years to complete. And the project generated a highly accurate sequence for over 90% of the genome. Almost 92% of the genome was completed by the end of Human Genome Project, which provided valuable insights into human biology. And the cost of initial human genome sequence by Human Genome Project was approximately $1 billion. Now, what is a shotgun sequence strategy? Shotgun sequencing is a lab technique for determining the DNA sequence of an organism's genome. So the genome is very big. So what is done in shotgun sequencing method is this involves breaking up of DNA into small fragments, which are then sequenced individually. And after they are sequenced, a computer program will look for the overlaps in the DNA sequences. It will reassemble the fragments in their correct order, and then it will determine the sequence of the starting DNA. So now during the Human Genome Project, shotgun sequence approach was also adopted. It played a crucial role. The DNA was broken into small fragments, typically around 150 to 200 base pairs in length, and it was done by using specific enzymes. And these fragments were then sequenced individually and then were individually using the automated DNA sequencing machines later. So there were also advancement in the Sanger sequencing techniques. The radioactivity was replaced by fluorescent tags. The nucleotides were tagged with fluorescence, and then reactions started to be performed in a single tube because all 4 nucleotides were assigned different colors. So there was no need of assigning 4 different tubes to carry out the sequencing process. So the sequencing was carried out later in single tube with the fluorescent tags, and it was read by the fluorimetry process. So the Sanger technique was also automated later, and it was faster than the original Sanger technique. Here in shotgun sequencing, once these sequences are aligned, a working draft of human genome was prepared, and after a working draft, the gaps were filled and the final sequence was ready. Now, about the first draft. So in 2001, scientists achieved a major breakthrough, which was called as first draft of Human Genome Project. There is quite a story about it. I think this lecture is not will not provide too much time to talk about this story. So this was a competition between the public group and Celera Genomics, which was a private company set up, which actually pushed the timeline. And because of that competition, this Human Genome Project, it was finished before time. So that's a story to tell up in some other time. So let's talk about this draft first. So in 2001, this draft was released. So the scientists, they worked together from different countries to decode the human genome, which is like our genetic instruction manual. It took many years of hard work to figure out the order of 3 billion building blocks that makes up our DNA. And this accomplishment opened up exciting possibilities for medical advancement and understanding how our bodies work. And it was a huge step forward in our knowledge of human biology and how our genes affect our health and trait. In 2003, the Human Genome Project achieved a major milestone by generating the first sequence of human genome. This was called as Human Genome Reference Sequence. So in collaborative effort involving 6 countries, 20 centers, and thousands of researchers, the Human Genome Project, it utilized brute force with Sanger DNA sequencing and extensive computational support because the data generated was massive. You can imagine data generated from 3 billion base pairs, like what kind of computer algorithms would have been used to store all that data, analyze that extensive data that was produced day and night, 7 days a week, and for approximately 6 years. This project sequenced about 1,000 bases per second. So this relentless dedication led to the successful completion of the project, uncovering the intricate details of the human genome and paving its way for the groundbreaking discoveries in genetics and medicine. So, however, this initial sequence was incomplete, with almost 8% of the genome remaining unknown. There were like gaps because of the technological limitations. Now, the limitations were, when we look at our chromosome, the centromere and the telomeric regions, they are highly repetitive. But here, when we utilized shotgun approach, we have used shotgun approach in sequencing the genome. So the genome was broken into very small pieces. They were sequenced and then they were aligned. So if you have repetitive sequence, you will not be able to align those. Sequencing is easy, but how will you align? The DNA is highly repetitive. You don't know when is this repetition starting and when it is ending. So by the time there were techniques that were developed which could read larger sequences, these highly repetitive end sequencing was nearly impossible. So there remained these 8% of the genomic gaps. And these certain regions with repetitive DNA sequence, they were exceptionally challenging to sequence accurately.
Next generation sequencing. Next generation sequencing has revolutionized DNA and RNA sequencing, providing significant advancement in genomics research. Now, unlike traditional Sanger sequencing, which analyzes one DNA strand at a time, NGS can simultaneously process billions of strands. And the Human Genome Project, it uncovered all 3.2 billion bases of human genome. This project started in 1990 and took until 2003 to complete 92% of the first genome. So, but in 2022, all the gaps got filled and the sequence became complete. So, in total, sequencing the human genome took 32 years. Now, in contrast, NGS can now sequence a person's entire genome in just one day. The speed enhancement is attributed to NGS's ability to analyze a large number of DNA samples simultaneously. And this breakthrough has transformed genomics research, enabling faster and most cost-effective analysis for genetic information. Next generation sequencing process involves several steps. First, samples are collected and the DNA and RNA is purified to ensure its quality. If RNA is being sequenced, it needs to be converted into DNA through a process called reverse transcription. Now, once DNA or RNA is prepared, first step is to create a library. This library consists of short fragments of DNA which are derived from the longer stretches of DNA from our sample. This is done by cutting the samples into specific sized pieces using either sound waves or enzymes. And then there are adapters, which are DNA sequence containing sequencing information, which are now adapters, which are DNA sequences containing sequencing information and sample identification. These are added to each end of the DNA fragments prepared in the earlier step. And then any unused adapters are removed. And this finalizes the library preparation. Now, depending on the specific requirements, a PCR step might now be needed to increase the amount of library. Otherwise, we proceed to the next step. Next step is attaching the library to the flow cell. So the main sequencing instruments which are used in NGS are specifically by Illumina. So it utilizes a method which is called as sequencing by synthesis. It involves using a DNA polymerase, a new DNA strand from the strand of interest. In this sequencing reaction, individual nucleotides chemically labeled with fluorescent tags are incorporated into new DNA strand. And when a nucleotide is added, it is excited by a light source. It emits a fluorescent signal that can be detected. And these specific signals, it will vary depending on which of the 4 nucleotides is incorporated. And this whole process, it takes place on a glass surface, and this glass surface is called as flow cell. On the surface of flow cell, there are small DNA fragments called as nucleotides, oligonucleotides, and these oligonucleotides matches with the adapter sequences from the DNA library we prepared in the earlier step. So to begin, the DNA library is separated into single strand by denaturation. These single strands are then added to the flow cell where they get attached to the oligonucleotides. Now this attached strand is called forward strand. Following this, a reverse strand is created while forward strand is washed away. So the result is this library becomes bound to the flow cell, which is ready for the further sequencing steps. Now, if the sequencing start now, the fluorescent signal will be too low for detection. So each unique library fragment, it needs to get amplified to form clusters. So this clonal amplification is done by PCR, but this all is happening in a single temperature, unlike the regular PCR steps. So annealing, extension, and denaturation all will happen by just changing the flow cell solution. So first the DNA binds to the second oligo in the flow cell. It forms a bridge. The strand gets copied based on the template sequence, and then these double-stranded fragments will get denatured. Denaturation will be done by using another solution to the flow cell, as already discussed. This copying and denaturation repeats over and over, and localized clusters get made, and finally the reverse strand gets cut. These strands get washed away and only forward strands are left for sequencing, and the same step is repeated for forward strand sequencing. Now once the sequencing is complete, the next step is to align and get rid of the bad reads. So the bad reads could be the clusters which have low intensity or unreadable intensity. So now the reads are mapped to the reference sequence. So the different reads will align to the reference sequence, and then there will be a lot of overlapping fragments also. So during the sequence alignment, the algorithm which are used, they will know that where will this single fragment read will belong. So the longer stretches of DNA or RNA, they get analyzed with greater confidence that the alignment is correct. So how is this NGS being used? So NGS gets used in wide variety of application in diagnosing cancer, rare diseases, treatment guidance for cancers, and many research areas from ecology to botany to medical science. Both DNA and RNA can be sequenced. It could be the whole genome or the transcriptome. It could be just the coding region called exomes of the DNA or the target genes in the DNA or RNA. All types of all types of RNA can also be sequenced, including the non-coding RNA such as microRNA, long non-coding RNAs. So in addition to cell-free DNA, single cells as well as methylation or protein binding sites can also get sequenced.
This is another advanced sequencing technique known as single molecule real-time sequencing. So this utilizes, This utilizes a high-speed moving camera and microscope to observe DNA polymerase molecules, which naturally replicate within our cells. To enhance the visibility, bright dyes of various colors are introduced with each dye that represent a specific DNA letter. So all the 4 bases, they are designated different colors. So this sequencing is also known as PacBio sequencing. This is a DNA sequencing technology that allows for direct observation and sequencing of individual DNA molecule. And this happens in real time. So in this SMRT sequencing, a single DNA molecule is immobilized on a solid surface, and a DNA polymerase enzyme binds to it. So this technique doesn't require amplification. The other techniques that we were discussing, like the Sanger sequencing and the next-gen sequencing, amplification is one of the most important step. We need to amplify our DNA to get enough signal so that the sequencing can be done. But here in SMRT sequencing, amplification is not needed. The sequencing is done in the real time. And the fluorescent labels, they emit unique signals, and it will depend on what type of nucleotide is being incorporated, and it can be captured in real time. So these signals, there is a specific camera, which is a specific microscope, which is equipped with a fast camera. It records the fluorescence in real time as the DNA polymerase incorporates nucleotide. This creates a continuous movie of DNA replication process, and it allows researchers to observe the sequence of the DNA molecule being copied. So the data which is obtained from SMRT sequencing, it provides long read. We do not need to fragment the DNA here also. So it means that the DNA fragment can be sequenced in a single read. So if fragmentation is not a part of this process, there is no need for assembly steps also. In the other sequencing method, we make fragments, we sequence them, and then we assemble them against the reference map. But here, because we are not doing any fragmentation, there is no additional assembly steps required. So this makes SMRT sequencing particularly useful for complex genomic regions, such as the repetitive sequences that we were talking about, the telomeric ends and the centromere ends. So making fragments and then aligning these complex sequences was impossible. This is the reason the Human Genome Project, which was completed in 2003, had gaps. But after having these kind of advanced technologies, which can read longer sequences, which do not require the DNA to be fragmented, we were able to close the gaps last year. So the SMRT sequencing, it offers advantages which is like high accuracy, long read length, and the ability to detect modification in DNA. It can even detect methylation. So if a DNA is methylated at certain point, the fluorescent signal that is generated usually will also change. So this feature, it makes it valuable for various applications like genome assembly, identification of genetic variants, and epigenetic studies. So it is worth noting that this sequencing techniques, it continue to advance with improvements in read length, throughput, and cost effectiveness. So it is making it increasingly powerful tool for genomic research. Now, other such technology is nanopore sequencing. This is an advanced DNA sequencing technique, also called a third generation sequencing technique, which involves passing DNA molecule through a teeny nanopore channel. It offers several advantages over the traditional methods. By applying electric current, let me open this video here. Yeah, so by applying an electric current across the membrane, DNA molecule can pass through the nanopore. There is a nanopore there in this design. And as the DNA molecule moves through the channel, there is disruption in electric current due to the individual bases. So different bases will have different change in electric current. The change is detectable. So this nanopore can detect which nucleotide has passed through it. So these are detected in real time and recorded as electrical signals. So these signals are then used to determine the DNA sequence. So it also has notable benefits. It can also read the long DNA fragments. It can help in analysis of complex genome and structural variation. And real-time sequencing allows for faster results because the other technologies, they'll take a lot of time in aligning the sequences, the algorithms, the computer, the bioinformatics. It will take a long time, but here we get faster results because everything is happening in real time. So this technique also has various applications. Now, this is an overview of how the sequencing has changed over the last few decades. This third generation sequencing techniques that we were talking about, such as PacBio and this Oxford nanopore technology, they offer independent amplification-free sequencing. Amplification is not required. These platforms can generate longer reads. So complex DNA can be sequenced. And now the complete human genome sequence. Now with these third generation sequence platforms, those complex regions which were highly repetitive, they were because there was almost advancement happening in every field, in genomics and technology, algorithms, computational biology. Scientists never stopped working even after the Human Genome Project came. They even started working hard. And now that we have advanced genomic techniques, advanced sequencing techniques, we were able to complete this human genome finally in 2022. So it started in 1990 and it was officially declared completed in 2003, but it wasn't until 2022 that this genome was fully completed. So it was completed using advanced sequencing mapping techniques, which were not available before when Human Genome Project came into picture. So the Human Genome Project, it mapped about 92% of the genome that we have already discussed. The remaining 8% included numerous genes and repetitive DNA. And this 8% was comparable in size to one chromosome. Because these regions were highly repetitive, it was impossible to sequence them using the shotgun approach for NGS. So using long read methods, the most difficult repeat-rich part of the human genome have been completely mapped. So this project was called T2T, which is short for telomere to telomere. It refers to the completion of human genome reference sequence from end to end. So over the past decades, the two new DNA sequencing technology emerged that we have already discussed about, the Oxford's nanopore sequencing technique, which can read up to 1 million DNA letters in a single read with a modest accuracy, while the PacBio, which we discussed earlier, PacBio's technique, it can read about 20,000 bases with nearly perfect accuracy. So the researchers in the T2T consortium, they used both the sequencing techniques that we discussed just now to generate the complete human sequences. So thousands of researchers have developed better lab tools, computational methods, strategic approaches to decipher this complex sequence. The 6 papers encompassing the completed sequence, they appeared in Science along with companion papers in several other journals. These are the references if anyone wants to go through. Now, while completion of the human genome was historic, and we learned a lot from it, we also learned that a single reference genome that's supposed to represent the average human actually misses a ton of important variation. Why? Because most of this genome came from the European race. So the people who were actually initially sequenced for the Human Genome Project, they were from one specific race, and it is not actually good to call it average reference genome, because it won't apply to most of the people from different ethnicity or the different ethnic group because of several variation in the genome. So completing the human genome reference sequence was only one part of the challenge, because human genome sequence, when we see as a whole, 99.8% of human genome is identical among all the humans. And this remaining 0.2%, it accounts for genomic differences that exist within human species. So population from different ancestors, they will have a specific genomic variation, and a single human genome reference, single human genome reference sequence cannot represent this diversity accurately. So now this Human Genome Project, which initially focused on sequencing genome of individuals from only the European descent due to logistics reasons and then the sample availability also, this limited perspective has led to a recognition that genetic diversity is vast, and it includes different races, and it includes different populations. So to capture the full range of genetic variation, subsequent projects such as 1000 Genome Project, it aimed to sequence genome from diverse population worldwide. So then came the concept of pangenome. So it has emerged because studying a single reference genome is insufficient to represent the entire human population. So a pan-genome, it includes collective genetic information from multiple individual and population, and it allows for more comprehensive understanding of diversity, the genetic diversity. So it also helps to uncover variation such as rare or population-specific genetic variants, which may have implications for disease susceptibility, drug response, personalized medicine, etc. And by studying a pangenome, we can gain insight on genetic variation that exists across different races and population, which can lead to more inclusive and representative understanding of human genetics. So this knowledge is crucial for advancing medical research, improving diagnostics, and developing targeted therapies that are cost effective for diverse population. So thankfully, the work has already begun and teams are contributing data sets to Human Pangenome Reference Consortium now. So this group is dedicated to bring equity to the reference genome. Okay, so why it is important, why sequencing, what is pangenome? So it is collection of genome from multiple individual. It can have similarities or differences in the DNA sequence. So the draft that is already prepared by Pangenome Consortium, it includes 47 genomes now, and they have plans to expand it to 350 genomes by 2024. And this expanded pangenome adds over 1,000 new gene mutations and nearly 120 million base pairs to the existing human genome reference, and which is a very valuable resource for future genetic research. So the publication of this pangenome in journal Nature has been done. It has been published in Nature along with the other related papers, and it is a significant step. Now, what do we do with all this massive information and data? What is the use of all of this? Personalized medicine. So right now, what is happening, what has been happening all over the world was there was a medicine. If someone has fever, there is a set medicine which we have to take, and it works for most individual. But there could be variations in gene, the variations in other complex genes, different genes, and the disease is not easy as just the fever, maybe some other complex diseases. And sometimes when one medicine works for one individual, it may or might not work for other individual in the same way. Maybe the dose required is higher, or maybe they require a different medicine altogether. So what is personalized medicine? Personalized medicine, it is also called precision medicine. And it is all about giving each patient the special care they need. It recognizes that everyone is different, and everyone may respond in their own way to treatment. So this is because like our genes, our lifestyle, where we live, how much money we make, it all can affect our health differently. So to provide personalized medicines, doctors use fancy technology. They analyze a lot of data. They collect information about patients' genes, their medical history. They also like to collect information about the way how they live their life, what they are exposed to in their environment. So then this information is used to understand the patient better and then make decisions about their treatment. So this personalized medicine, it allows doctor to create customized treatment plans. Instead of using one size fits all approach, they pick treatments that are most likely to work for each patient. This way, they can increase the chances of success while avoiding treatment that might not work or cause problems, because sometimes medicines also cause side effects. So those can be avoided. So by using advanced technology and analyzing data, doctors can make smarter choices. They can give each patient the best care possible. And personalized medicine is exciting because it can improve the patient outcome. It can make treatments work better and even save money by making it more efficient. So hit and trial and hit and trial and seeing if the treatment works, and then shifting to another medicine, another treatment plan. It can save a lot of trouble, it can save a lot of time, and lot of agony. And of course, it will be cost effective if we from the beginning know that which treatment is going to work for which individual. So personalized medicine will mean that doctors or healthcare providers, they'll focus on giving care that is specific to each person. They will understand that each person is different, and this person might respond differently to the treatment. So they take into account things like genetics, the gene that you inherit from your parents. They also take into account your lifestyle, the environment, and all the socioeconomic factors, and then prescribe you medicine, and or the treatment plan can be devised based on your individual personal conditions. So it uses advanced technology, data analysis, and all this data that we were discussing earlier, all these sequencing techniques. This data can be collected. There are databases, and these databases are growing every day with more sequencing, more number of people going for genome sequencing, whole genome sequencing, and other kind of sequencings. This database is growing, and our understanding of how a gene or a set of genes are working is also getting better with each day. And precision medicine has a way to go with all these new information getting here, and more people being aware of the advances in the treatment. So this was just an overview. The other technologies, in summary, we have already discussed that it focuses on individual care. Each patient is unique, and it considers different factors. Now, what is circulating tumor cells? This is also a part of precision medicine. Circulating tumor cells, these are the cells that break away from tumors, and they travel through the bloodstream. So these can be isolated from the patient's blood, the patients who are suffering from any kind of tumor or cancer. So the DNA, RNA, and the protein in these cells, they are isolated to understand their unique characteristics and predict if they have ability to cause metastasis. So when these circulating tumor cells are isolated, their DNA is isolated, it is sequenced, and then it is analyzed against the reference to see what kind of characteristics these tumor DNAs have, what kind of treatment they will respond to, and what kind of treatment will not work, how the disease prognosis is. Because this is a very simple test, only you have to draw blood. So if the patient is given any treatment, you can also, the doctors can also, monitor it time to time, how the treatment is going on, if the patient is responding to the treatment. So these circulating tumor cells, they have unique advantages over the other methods. They can help us guide the personalized treatment. They can help us understand how the cancer spreads. They can also help us with the early detection. Sometimes when the cancer is detected, it is usually too late. The cancer is already causing the symptom. Maybe it has metastasized to some other place. So the treatment then becomes very complex. So this is a simple blood test. If we can get it into not routine, but if we are predisposed to some genetic condition which makes us more vulnerable to certain kind of diseases, say cancer, we can go for liquid biopsies. So this test is called liquid biopsies. We just have to give our blood and see if there is presence of any circulating tumor cells. If there is, individualized treatment plan can be devised for this patient, and they can benefit from it and save the pain of the future chemotherapy and other exhaustive treatment plans which the cancer patients undergo. So here NGS comes into picture. NGS can unlock valuable insights into the genetic makeup and the changes of tumor. They can identify the tumor and provide important clue about the cancer. They can help predict if the cancer will come back. They can also track its progression. They can assess how well the treatments are working. So these circulating tumor cells, they are also being used to create models that mimic how patients will respond to chemotherapy, and like it can also be used to test new treatment plans. So this will help the doctors understand which treatment plan may work best for what individual. So like excitingly, this liquid biopsy, it has been approved by FDA for cancers of breast, prostate, and colorectal cancer. So this is already going on. This test is called liquid biopsy. It offers this non-invasive way to gather important information about a patient's diagnosis, its prognosis, and the prediction of treatment outcomes. And it's truly exciting that a simple blood test can now count CTCs in specific cancer. They can provide non-invasive information about diagnosis and the treatment possibilities. So this is a very important example of personalized medicine approach. Another application is non-invasive prenatal testing. So this is a screening test which is performed during pregnancy to evaluate the risk of certain chromosomal condition in the fetus. So if the family is predisposed to some condition, if they already have a child with some kind of anomaly, or any of the parent is a carrier for some disease, in these conditions, this test can be taken. It involves analyzing cell-free DNA. So what happens, like a fetus, it also there in the mother's blood, the cell-free DNA is found from the baby, from the developing fetus. So NIPT, it offers a non-invasive method wherein the patient's blood is taken, and from the maternal bloodstream itself, this fetal DNA can be analyzed, and there is no need for the more exhaustive tests like amniocentesis, which also have some risk of miscarriage. So after drawing blood from the mother, NGS can allow for a comprehensive examination of fetal DNA. It can detect even minor genetic variations or anomalies. And by comparing this information with the established reference genome, NGS can help us identify the deviations and may also indicate the presence of if the baby has a chromosomal abnormality or any other abnormality. So this provides the expectant parent with valuable information about their baby's health so that they can make informed decision. This test is safe because it is just a blood test. It is non-invasive. And with high accuracy and reliability, this NIPT using next generation sequencing, it has revolutionized the prenatal care, offering a comprehensive and convenient approach to assess the genetic well-being of the fetus. Now, the advanced genomics about cancer, since we have talked about cancer, what else these advanced genomic techniques can do? So next generation sequencing, it is a powerful tool in studying and understanding various diseases which beyond cancer. So there are some examples, and NGS applications can help in different disease area. So one is genetic disorder. So advanced sequencing techniques can be used to identify disease-causing mutations in individuals with Mendelian disorders, which are the inherited disorders such as cystic fibrosis, muscular dystrophy, and Huntington's disease. And it can identify rare genetic variants which are associated with rare disease. It can aid in diagnosis, and it can facilitate in development of targeted therapies. And the second one is infectious disease. So it can rapidly, the advanced genomic techniques, which can include NGS, it can rapidly and accurately identify infectious pathogens. It can identify bacteria, virus, fungus, parasites from the clinical samples, and it can help in diagnosing and managing infections. That can save a lot of time. And also antimicrobial resistance. So it can detect and characterize genetic markers which are associated with the antimicrobial resistance, and which can assist in the selection of appropriate treatment options. Third is neurological disorders. So NGS may allow the identification of genetic variation which are associated with neurodevelopment disorders in children, such as autism spectrum disorders, or intellectual disabilities. It can also help in uncovering genetic risk factors and mutations which are associated with the neurodegenerative disorders such as Alzheimer's and Parkinson's disease. Next is cardiovascular disorders. So the genetic variants which are associated with inherited cardiac conditions, including cardiomyopathies, arrhythmias, or congenital heart diseases, they can also be identified using NGS. And identification of genetic variants which are associated with the condition, which are characterized by high cholesterol levels, increased risk of cardiovascular disease, can also be done. And then detection of genetic variants which are responsible for hematological disorders like hemoglobinopathies, such as sickle cell anemia and thalassemia. They can help in early diagnosis and maintenance management of these diseases. These advanced genomic techniques can also be used for genomic profiling of leukemias, lymphomas, which can be used to identify specific genetic alteration. And this can help in guiding treatment decisions, which is very important because hit and trial takes a lot of time, and sometimes patients do not have that much of time to do trial and error and change medications when one fails. So having a treatment plan, a solid treatment plan from the beginning, can save very important lives. So guiding treatment decisions and monitoring minimal residual disease. So these are the advantages of personalized medicine. So identification of disease-causing mutations, improved diagnostics, improved accuracy in diagnosis, guiding personalized treatment approaches like across multiple disease areas, and this can ultimately lead to better patient outcomes. Now, a little bit about Strand. So this is a company I'm working with for past few months. It is a CAP and NABL accredited laboratory, and it is India's first laboratory to get both the recognition for NGS testing. So they offer comprehensive genomic services to the scientific community with lab-hosted technology platforms. So Strand Life Science Services can help in disease understanding and management by providing personalized information on disease risk, drug response, and treatment options, whatever we were discussing today, but whatever we have discussed on the personalized treatment options. So by analyzing genomic data, disease-causing mutation can be identified, and personalized treatment strategies can be developed, which can ultimately improve the patient outcomes. So the first thing Strand does is genomic profiles. They offer comprehensive genomic profile service that can help identify genetic mutations and variations which are associated with various diseases. By analyzing the DNA sequence of an individual, it can provide information of disease risk, drug response, and then the personalized treatment options. Second is clinical diagnostics. They offer a range of diagnostic tests for inherited diseases, cancer, and infectious diseases. And they use state-of-art technologies to provide accurate and reliable results, helping doctors make informed treatment decisions. Now, the more important part is bioinformatics analysis. Like when it comes to genomics and advanced sequencing tool, this is very important aspect, that immense amount of data is generated. So it is truly mind-boggling. We are talking about the data in the range of terabytes or even petabytes. So dealing with such massive amount of genetic information is no easy feat. So it requires powerful computers, it requires smart storage solutions, and carefully designed analysis process. Special tools and algorithms are needed to handle this data explosion. So it is like trying to solve a puzzle with billions of pieces. So, but with the right expertise and technology, this secret which is hidden within the vast sea of data can be unlocked. So the third service that Strand provide is bioinformatics analysis. They provide bioinformatics analysis service on the advanced data analysis and interpretation of genomic DNA, which is very important. What will you do if you have massive data that you cannot read, that contains only the bases, the series of numbers, A, T, G, C, T? If you won't be able to understand anything. That will be like huge, enormous data. So they have important tools. They have large scale genomic data sets that identify disease causing mutation and which can help in develop personalized treatment strategies. And fourth is the software solution. They also offer software solution for genomic data analysis, its interpretation and management. So their tools are designed to help researchers and clinicians to analyze and interpret genomic DNA efficiently, leading to faster and more accurate diagnosis and treatment decisions. This is an overview of what all services they provide. Now, the range of tests which are offered by Strand Life Sciences, they are from rapid point of care tests to specialized molecular tests in various fields such as oncology, genetics, fertility, mother and child care, infectious disease, and algorithm wellness. So the summary of what we have discussed so far, the Human Genome Project. Let's start with the Human Genome Project. So it started from 1990, and the first Human Genome Project was deemed complete in 2003. And the technique that they utilized was they mapped the genome first, and then they sequenced the genome. Sanger DNA sequencing was utilized by Human Genome Project for the sequencing process. And sequencing posed significant challenges because of the repetitive regions, because of how large the genome is. And all the obstacles in the repetitive DNA, they were later removed in 2022 when the genome was complete, telomere to telomere. All the repetitive parts of the chromosomes were also sequenced. So in 19 years later, after the publication of Human Genome Project, a truly complete T2T human genome was finally produced. Now the summary of personalized medicine. So personalized healthcare is also known as precision medicine, and it takes into account individual patient for their unique characteristics. It analyzes genetic makeup, biomarkers, clinical data for disease prevention, diagnosis, and treatment. So it emphasizes on prevention and early intervention rather than a reactive approach. Drug prescribing can be improved by minimizing trial and error. And CTC and liquid biopsies for cancer detection and treatment are being used lately, and they are very good techniques for devising personalized medicine and saving a lot of pain and time and cost for the cancer patients. And NIPT with NGS is also a very nice personalized and accurate assessment system. NGS has been instrumental in various fields, including cancer genomics, infectious disease outbreak, and rare genetic disorder diagnosis. Thank you so much for your attention. I know it has been a really long lecture, but I hope I could help you understand some of the aspects of genomics. Thank you very much.
Dr. Damodar, if you want to add something to NGS, then please.
He has explained every single thing, even in very, very nice way. And about the precision medicine, the requirement of NGS in oncology, the diagnosis, the treatment planning, and how a person is going to respond. The most important thing for all this technique and technology development is how to save life and how to improve life of an individual. And she focused on every single aspect. So how we can help to provide best possible benefit for all living beings? This should be our aim and focus. So very, very nice lecture, Dr. Shweta. Congratulations. And I think there are some questions available in the chat box and something that can be answered by Dr. Shweta very easily.
Thank you very much, ma'am, for your such an informative session. And now the forum is open for question answers. So students are requested to post their questions as well as they can raise your hands so that we can give the rights to unmute and they can solve the queries. Okay.
Dr. Ritesh, you are
Yes,
not
now you
audible.
can try.
Yes, yes, now you are. There are some questions available, like initially there were no standard sequence available for human. Then how they decided to decided which fragment come first. I think it was explained in coming slide already. I hope it was clear. If it is not, then it can be answered again. Shweta?
Madam, you can try once again. Now you will be able to unmute yourself.
Yeah, I was trying earlier, I could not.
Hmm.
Okay, so I missed this part. Human genome was mapped first, like it was not like initially fragmented. The first approach for human genome sequencing was to identify the landmarks in the genome. So first the landmarks were identified and a map was made before fragmenting the entire genome. And later, when the small fragments were made, they were aligned as per the map. So I did include it in the summary, but not in the presentation because it would have made it really bulky. So the mapping first part came first. So when we have a map of a, like, say, for example, if we have a circular DNA, let me open a whiteboard if it's possible here. Actually, I can't. So let's understand it this way. So there is a map, let's talk about a pentagon, which has 5 sides, and each sides let us place one marker of different colors. So initially we would identify what color of markers those are, and then we will divide this pentagon into 3 parts. Then it will be easy to, detach those 3 parts and attach back because it is a big pentagon. Next, we will break it into maybe 7 parts. And based on the information we had earlier, we'll be able to again put it back. So this way, a map of the entire genome was created first with the big landmarks. And after this map was formed, the sequencing was done. So the human genome sequencing started with mapping the genome first. I hope I answered it. Okay, the second question is, does this liquid biopsy detect benign tumors? What if the tumor resides are? So tumor will not reside in blood. Tumor, if it is a solid tumor, it will be in different sites. So, but wherever the tumor is, the tumor cells are also dying. So these circulating tumor cells sometimes are dead cells, sometimes live cells break free from the site. So our blood, mostly it is now known that most kind of cancers, let's not talk about benign, I'll talk about benign tumors later. Let's talk about the active solid tumors, which are cancers first. So any tumor of like specific lineages, mostly the breast cancer, the prostate cancer, and the colorectal cancer, it has been found if a patient is suffering from these kinds of cancers, their blood has circulating tumor DNA. So what happens is, because these tumors are losing some cells into the bloodstream, these can be captured through a simple blood test. So can I go on? Okay, so these are not already in the blood. These are being, released in the bloodstream. So their site of organ is something else. This is how metastasis also happens. No, when an active cancer, it decides to metastasize. If it has conditions to metastasize, it breaks free from the original site. It goes into the bloodstream or the lymphatic system, and then it will reach a new site and then develop into a different kind of cancer. This is how metastasis occurs. It is the same way. So all the cancers do not metastasize. To have metastasis, there are certain prerequisites these cancer cells need to have. They need to, not all cancers metastasize, some do. So the similar things happen when there is a tumor growing. It will release some of its cells to the bloodstream. Whether it is a metastasizable cancer or not, some of the cells get into the bloodstream, and these cells can be captured and utilized for liquid biopsy. And now if we talk about benign tumor, usually benign tumors have a very defined boundary. They do not grow that fast. So if they are not growing that fast, if they do not invade a nearby blood capillary or a blood system, they won't get into the blood. So if they have access to blood, if they can detach from the cancer site and get into the blood, they can also be get detected into the blood system through liquid biopsy. But usually benign tumors, they stay within a boundary, within a defined system, and they do not invade blood system. So these kind of tumors, they are difficult to find, detect through the this liquid biopsy. Now, how is a fetal DNA differentiated from maternal DNA? So there are techniques to see, like when a test is being done for maybe a prenatal testing, the gender of the mother, what gender the mother is. So once, if the fetus is a different gender, say the fetus is male, you can definitely see. But when you do the sequencing, you will get two set of the genome. One is definitely mother, other is the child. So if the gender is male, you do not need any other testing. But if the gender is not male, if the fetus is also a female, then there are additional tests that can be done. The DNA sequence of other cells from the mother can be checked, like the fetal DNA concentration in blood will be very less. There are different kind of blood cells which are present in the system, right? So mother's DNA will be much higher in concentration. Mother's DNA, mother's genetic material will be much higher in concentration, because all the blood cells except RBCs, they will have, like RBCs and platelets, they'll have nucleus, and then you can isolate the DNA from mother's blood, and then the smaller part of DNA concentration, you would know that it has been secreted from the fetus. Any other questions?
Yes, any other questions from students? Thank you, ma'am. Thank you for the satisfactory answers. On behalf of Ganpat University, I express my deep gratitude to ma'am for accepting the invitation and giving her presence.
Thank you so much for this opportunity.
I request Dr. Dhaval Prajapati to present a statement of gratitude to ma'am as a token of respect and appreciation. Sir, please. Ma'am, please accept it.
Thank you so much.
Now I request Dr. Hardik Shah to deliver a vote of thanks for today's esteemed speaker.
Good afternoon, everyone. On behalf of Ganpat University, Mehsana Urban Institute of Sciences, Department of Microbiology and Medical Lab Technology, I would like to express our heartfelt gratitude to all of you for joining us today in this informative and insightful webinar on genomics and next generation sequencing. We are honored to have had a privilege of hosting esteemed speakers, dignitaries, and participants from various fields and backgrounds. First and foremost, I would like to extend our sincere appreciation to our distinguished speaker, Dr. Shweta. Her expertise and profound knowledge on genomics and next generation sequencing have provided us with the valuable insights and deeper understanding of this cutting edge field. We are grateful for her time, dedication, and enlightening session she delivered. Thank you, ma'am. In addition, we also extend our heartfelt thanks to our authorities, Dr. Ganpat Patel, our beloved Dada, Patron-in-Chief, Ganpat University, Dr. Mahendra Sharma, sir, our Director General, Ganpat University, for their visionary leadership and continuous support. In addition, I would like to also thank Dr. S.S. Pancholi, Executive Dean, Faculty of Pharmacy and Faculty of Science, for his guidance and encouragement. Dr. Amit Parikh, sir, Dean, Faculty of Science, Ganpat University, and Principal, Mahesana Urban Institute of Sciences, and Michelle Neil Rami, Coordinator of Department of Microbiology, for their unwavering support and mentorship. We also like to express our gratitude to all the team members who had worked tirelessly behind the scene to make this webinar successful. Their dedication, hard work, and attention to detailed ensured the smooth execution of this event. A special word of thanks goes to all the participants who joined us from different parts of India and the world. Your active participation, insightful questions, and enthusiasm had made this webinar truly engaging and interactive. I hope that the knowledge gained from this session will prove beneficial in your respective fields and endeavors. Last but not least, we would like to extend our appreciation to any other individuals or organizations who have supported us in organizing this webinar. Your contribution and collaboration have played a crucial role in the success of this event. In conclusion, we are deeply grateful to everyone who has contributed to this webinar on genomics and next generation sequencing. Your presence, support, and engagement have made it memorable and enriching experience for all of us. Thank you once again. We look forward to your continued participation and support in our future events. Have a wonderful day. Thank you.
For the same, I would like to invite Dr. Ritesh Arya to conduct the quiz. Please, sir.
Now, the most awaited movement for all the participants is here. We are moving towards our Menti quiz, today's quiz. So I request all the participants to open menti.com. I have provided the link in the chat box. I request all participants to please press on this link, that is menti.com. Now, all of you are requested to enter today's quiz code, that is 42947.